I hope every one of you had very good lunch and a networking session and a lot of new people. But we continue our lectures, and the next talk is very important for me because it will be given by Mireya Plass, and she came from the Berlin Institute of Systems Medicine, which is in MDC in Germany. And she came from the laboratory of my beloved Nikolai Vostoyevsky. And the talk will be dedicated to single-cell sequencing of amazing creatures, planaria, who tend to live forever. So, Mireya, you're very welcome, and enjoy, Mireya.
Okay, thank you. So thanks for this very nice introduction. I hope that the talk is as exciting as you promised it will be. Yeah, so my name is Mireya, and today I would try to explain you this project that I was working on for the last more or less 2 years, and that we recently published. But first I would like to make an introduction of how do I see that is single-cell transcriptomics. So I think that nowadays we are doing lots of things with single-cell transcriptomics, but in some way it's just another tool to study biology. And basically what it's giving us is the opportunity of studying biology at a much higher depth. So if we think about just an organism, an animal, like this typical one, you know, with conventional RNA-seq, we are only able to study different tissues, maybe compare, like if you treat your animal or your patient with a drug, what are the effects in general. Or you can get cell population averages, like get a biopsy of a brain or something like that and try to figure out what is in there. But with single-cell transcriptomics now, we can do much more than that, right? So as you have heard in the talks this morning, we can, okay, this works. We can identify and characterize, like this, yeah. We can identify and characterize different cell types. And these cell types that are these different clusters that we have here, they represent different cells or cells at different maturation stages, like progenitors and differentiated cells and stem cells. But on top of that, what we can do is try to infer the relationships between these cells. So we can try to understand whether, for instance, these cells here originate from these cells here or from these cells here, or what is the relationship between different types of mature cell types. So I would try to explain you all this today using as excuse the paper that we recently published, in which we try to do all this for the Planarian Schmidtea Mediterranea. And basically, my talk will be divided in 3 parts. In the first part, I would explain you how we use dropseq, or I would say that you can use any kind of single-cell transcriptomics to build a cell atlas. The second part would be how we can work with this cell atlas to try to infer the lineage relationship between the cells. And the final part of the talk would be how we can use this single cell to do something more than just building analytes and finding lineages. So try to understand complex biological processes. And in our case, we will try to study regeneration. So before going to any of these topics, I would like to explain you why we work with Planarians. And I think that Nika already said it, but so we can say that Planarians are immortal in a sense, because they actually can regenerate all body parts. So they continuously regenerate everything. And basically, that what means is that if you take a planaria and you cut it in pieces, each of these pieces is going to generate a full animal in a matter of 10 days. So as you can see, each of these pieces just becomes a whole animal, tiny one, but complete in 10 days. And why is that? So the fact is that planarias contain lots and lots of adult pluripotent stem cells. So this is a section of a planaria. Sorry that the slide is black, otherwise you cannot see anything. I hope that you see. And basically, these are like the main organs of planarias. We have the eyes, the brain, the pharynx, that is the mouth, the gut, the verticals, the skin, and all these green little dots that you see here are these adult pluripotent stem cells that give Planarians these awesome regeneration capabilities. So therefore, we thought that was the ideal animal, so we are done, ideal animal to study stem cell differentiation, because basically, if we get a sample of cells from Planarians, we are going to have a large pool of stem cells, these neoplasts. We are going to have all possible differentiated cell types in the animal, but on top of that, we are going to have progenitors at all possible differentiation stages. Therefore, it would allow us to reconstruct the lineage of this animal just by sequencing the adults. Okay, so now let's start with the project. So we want to build an atlas, right? So what we did was to take these cells, dissociate them, and do dropseq, as it was explained earlier this morning. And so I'm not going to go with details, just the fact that you get these microparticles with the cells, and you can capture all the RNAs within the cells. And basically, in the end, what we get is this. This is a digital gene expression matrix that is going to summarize the expression of all the genes in all the cells that we have identified. And then we need to make sense of this. So what do we have here? What does it mean? In our case, what we did was a t-SNE, like the ones that we showed before. But basically, what you get is this. So each thought is a cell. We don't know what it is. We don't know what it does. From this plot, the only thing that we can say is that the cells that are close by, they are similar. And the ones that are further away, we have no idea what they are. So the first thing that we need to do is to use some clustering technique. If it appears, great. So we need to find a clustering technique so that we will be able to define groups of cells that are similar. So when we did this, but we got something like this. So we got 51 different clusters that still we have no idea what they represent or what they mean. And here we need to invest a lot of time and use previous knowledge, biology, to try to identify what are these cell types. And that's what we did. We compare our gene expression profiles from each of these clusters with known markers for different cell types in Planarians to try to figure out what they are. And then we get something like this. And what we found out is that actually our sequencing contains cells belonging to all the major lineages known in Planarians. So we have neuronal cells, we have muscle cells, epidermal cells, gut cells, and a giant pool here of stem cells in the middle. But now this is still a computational prediction, so we need to try to figure out whether this is true. As we saw before, we can look at marker genes and see whether these muscle cells express genes typical from muscle. But we can also, in Planarians, validate that in vivo by doing in situ hybridizations. So basically, what we can take is these marker genes. For instance, in this case, this is a gene that is specific from cholinergic neurons, and we can stay in the planaria and look where this gene is expressed. And in this case, okay, here you have to trust me a bit, but what we see is that it clearly marks the brain and the nerve cords that this animal have. And in this way, we validated many of our cell types. Many of them were known already, so they have been previously described. Some of them were very rare, so maybe less than 10% of the cells of this animal are like this, as for instance, this epidermis dorsoventral, that is, you see, is this tiny cluster here. And it's only expressed, so it's a kind of epidermis that it's only expressed in the separation between the ventral and the dorsal part of the planaria. And it's only these few cells here in the side that they are lit. And we even were able to identify new cell types that were not previously characterized molecularly or in any other way. As for instance, these two different types of parenchymal cells that have, as you see, very different expression patterns. So, but the most interesting part of all of this is that we can not only distinguish cell types, but also discriminate close related cell types. As for instance, in this case, we identified markers for goblet sets and phagocytes that are the two main cell types in the intestine of Planarians. And in this fluorescent in situ hybridization, you can see that we have these red cells, the phagocytes, the green cells, and here in this co-staining, what you can see in the overlay, what you can see is that they are totally separated. So finally, what we can say is that actually this is quantitative. So what we did was to compare the clusters that we identified with our dropseq, grouped in different groups according to the type of cell they belong to. And we compared this data to some analyses that were done using microscopy in the 80s. So people in the 80s took these animals and counted by hand how many cells of each type they had. And this is the comparison. And as you see, it's fairly similar. So we have similar proportions of stem cells, epidermal cells, neural cells, parenchymal muscle. So we can not only make an atlas, but it's also a quantitative atlas. So what we are getting with the dropseq reflects what we have in reality. So the conclusion for the first part is that we can use single-cell transcriptomics to actually make a quantitative atlas that we can validate, and we can characterize the cell types that we have identified. So now let's go to the second part of the talk. So how we can reconstruct the cellular lineage of these cells. So in this project, we kind of got this idea that we have built a recipe on how to make this lineage reconstruction. And the idea of this recipe is that we try to combine as many different types of information as possible to try to make a robust prediction. So the first part that we did was to use sequencing specific cell populations so that will help us guiding this tree in some way or validating the overall trajectory. The next part that we did was to develop a new computational lineage tree reconstruction that would try to connect these different cell types that we have. Next, what we did was to use gene expression changes to try to assess how whether these predicted lineages were correct. And in the end, what we did was to use RNA velocity, that is a recently developed method that tries to predict the future state of individual cells. And by combining all these different data sets together, what we can do is develop a lineage tree that is robust and incorporates information from all these different methods, so we are reducing the probability that is totally wrong. So now I will explain you each of these parts separately, and let's see whether you follow me, the idea. Because we apply it for Planarians, but what we believe is that actually this can be used for anything. So what we did in the first part was to sequence these specific populations. In our case, we sequence these cells that are called X1 cells. These are double DNA content, so it means that there are cells that are about to divide. And we know in Planarians that this only happens in stem cells. And when we sequence them, what we see is that they are here in the tisney. If you remember, this was where our stem cells were. So that's pretty cool. So the second thing that we did was to sequence histone to be resistant cells. So a histone is a protein that is essential for cells when they are dividing. So if we remove it, what happens is that the cells die. And if you wait a few days, what happens is that they differentiate. So basically, we get rid of all the stem cells and progenitors. And all the remaining cells, what they are is terminally differentiated cells or late progenitors. So if we take this information and combine it with the information about marker genes for stem cells, we can do a simple principal component analysis. And what we see is that we can separate the stem cell clusters that are here located in the center in gray. In between, we have progenitor clusters that are all these yellow clusters that are marked here. And in blue, we have all differentiated cell clusters that correspond to the most peripheral clusters in our tisney. But still, this information, because this is a tisney, we cannot rely on the relationship between the cells, so we need to do something else to figure out how they are connected. So for this, we need to develop a method that will take us from these clusters to a tree that would relate our cell types. So we did this in collaboration with Fabian Types Lab in Munich that developed this algorithm that is called PAGA. That the main idea is that it goes from this n-dimensional cell graph, KNN graph, where we have all the gene expression. We can connect these cells to the similar cells. We can apply some clustering on top, and then connect these clusters based on the connections that we had in the original single-cell graph. And after removing some of these non-relevant links, we get like an abstracted tree that represents a possible lineage tree for this animal, or the most likely one. When we did this with our data, what we got was something like this, where we have all these thin lines are the possible connections between cell types, and these thicker lines, it's the most likely predicted tree. And what is interesting from this tree is that we have all differentiated cell types are here in the edges. They are connected to progenitor cell types, and they are rooted to a single population of stem cells. So actually suggesting that this population is the one that is giving rise to all these cells. So we validated this by using our previously computed experimental data. So we see that our double DNA content stem cells are mostly located here in the center of the tree, and that our differentiated cells are mostly enriched in the end part of this tree. So overall, suggesting that the topology between this tree is correct. But with this tree, we not only get these connections, but we also get a pseudotemporal ordering of the cells. So we can sort our cells and figure out how they differentiate in a progressive manner. But we also need to validate whether this was correct. So, and we did this by taking one of the clusters that we have, goblet cells, and we found out that there were markers marking two different populations within this cluster, this Bruchot and ADB, that they represent cells that are more close to the central neoblast cloud or more peripheric. When we take the cells of this cluster and we order them according to pseudotime as here, what we see is that at the same time that the stem cell markers drop, as in this case, this is METW1, that is the most classic marker for stem cells in Planarians, what we see is that these two genes peak at two different times. So we have first picking this marker of what we thought were immature cells, and later this marker of mature cells. But on top of that, what we could do is validate it using, again, in situ hybridization. So in Planarians, we know that the stem cells migrate to their finite location while they are differentiating. So according to this, what we saw is that these cells expressing these immature markers, Bruchot, were further away from the lumen, here, than the mature cells that are closer. And here you can see it in the overlay that you have red cells somewhere further away than the green cells. So now that we know that the ordering within one cluster is correct, now we have to figure out whether the connections between clusters are correct. And for this, we use gene expression. So the idea is that, for instance, if we take a single lineage like this one, where we have the stem cells giving rise to progenitors and giving rise to mature phagocytes, so we can take these cells, we can order them according to pseudotime, and if this is correct, what we would expect is that there will be a gradual change in gene expression from the stem cells to the mature cells. And actually, in this case, that's what happened. So we took marker genes for all these populations, and we looked at how they express. And what we found is that there are some groups of genes that are highly expressed in the stem cells and whose expression drop during the differentiation, while there are other genes that are no expressed, basically, or very lowly expressed in the stem cells, and their expression increase in progenitors and its maximum in the mature cells. But in all the cases, this was not like this, and in some cases, what we found is that the prediction was incorrect. So for instance, in this case, the prediction that we got with PAGA suggested that we had some progenitor cells that would give rise to muscle body and then muscle pharynx, two different types of muscle cells in Planarians. And what we saw is that in the same way that we had some genes that were highly expressed here for the stem cells and their expression drop, what we saw is that, for instance, specific markers would increase their expression kind of simultaneously in these two cell types, suggesting that instead of having a linear progression, what we will have is a bifurcated path, suggesting that actually both body muscle and muscle pharynx would come from muscle progenitors. So finally, what we did to complete this lineage tree was to use RNA velocity. So this method that was developed by the Karchenko and Linnerson lab was published recently this year. What it tries to do is predict the future transcriptomic state of a cell. So if we think about the life of an mRNA, that we get the DNA, we get it transcribed, then spliced, and then we get mature mRNA, and finally it gets degraded. We can assume in a simple model that in the steady state, the amount of RNA that we have in a cell is the same as the amount that we transcribe minus the part that we degrade. But when we want to increase gene expression, what happens is that transcription has to be higher than degradation. And in contrast, when we want to decrease gene expression, the transcription has to be smaller than the degradation. So considering that we can estimate the amount of transcription by the amount of spliced mRNAs and we can get the amount of mature mRNA from the splice reads, what we can do is try to plot for each cells these unspliced reads versus spliced reads, and try to identify the relationship between them, that is this gamma, that would represent the degradation rate. And by using this, using a simple formula, what we can do is to try to predict the future amount of mRNA in a cell, considering only these three parameters. So the amount of unspliced reads, the degradation rate, and the amount of spliced reads. So if we apply this to our data, what happens is that what we can have is one cell, and now we can project this future cell by looking at the most similar cells in our t-SNE. And the resulting plot is something like this. So this is the same plot that we had before, and now we have all these little arrows that, as you can see, are here pointing towards the stem cells, towards all the different lineages that we already described. So suggesting that actually these cells are giving rise to all the differentiated cell types. And if we analyze this and we see like where all these arrows are ending or where they are starting, what we see is that most of them are starting here, where we know that we have our stem cells, because we sequence them separately, right? These X1 cells. And we find all the differentiated cells here in the edges. So therefore, what we can do now is put all these things that I told you, our prediction, our experimental data, the gene expression changes, and the velocity, and we can make a tree that looks like this, and actually contains information from all these methods. So we have these thin lines that are the predictions from PAGA. We have these red ones that are the ones that we corrected based on marker genes expression. And then we have all these thick lines that are all those relationships that both PAGA and Velocito support. And on top, we have like the prediction of whether they are differentiated cells or progenitor cells based on our experimental data. Together, building this very robust tree that combines all this information. But the question is, why do we want a tree like this? So what can we do with it? Well, so the first thing that at least we thought of doing was try to figure out of identifying genes that are involved in the differentiation of these stem cells. So for this, what we can do is like take all the cells from all these lineages and make like a matrix and do some clustering so that we identify genes that change simultaneously in many, many cells. And then try to do some kind of easy representation so that we can understand what it means. And in this way, what we found is that there are some gene sets that are only expressing one cell type. For instance, we found this gene set that has 37 genes. Most of them are stem cell markers, and what we see is that they are highly expressed here where we have the stem cells, and their expression drops during the differentiation of most of the different lineages. Or other gene sets that are turned on during the differentiation of one specific lineage, as is this case, that has genes that are specifically expressed in the neuronal lineage. Then we have other gene sets that are involved in the differentiation of several lineages, as for instance, these two genes that are expressed both in the differentiation of the gut and the epidermal lineages. And in the end, we have genes that are expressed in multiple lineages at the same time, so they seem to be involved in the differentiation of stem cells in general. Yeah. And then, so I hope that I have convinced you that we can use single-cell transcriptomics and that we have developed a new method to reconstruct the lineage way in an unbiased manner, that we have validated the differentiation paths using gene expression and RNA velocity, and we have exploited the lineage tree to identify gene sets involved in stem cell differentiation. So just to finish, I would like to explain you how we can use single-cell transcriptomics to study complex biological processes as the case of regeneration. So as I told you before, Planarians are able to regenerate all their body parts, right? So that's why we set this study in which the basic idea is like we take a planaria, we cut them in pieces, yeah, and discard their heads. And then what we did was to sequence these pieces at different time points during regeneration. So just after cutting, 2 days after, and 4 days after. As you can see, they give populations of cells that are in different locations on the t-SNE. And now, what can we do? So the first thing that we can do is like take all the cells from these clusters and try to identify cell type specific responses during regeneration. So for instance, these are two of the clusters, the main neoblast cluster, the main stem cell cluster. and the body muscle cluster. And here what we did was to pool the data of each of these clusters as if it was conventional RNA-seq and identify signatures present, like genes that were overexpressed or I don't know how to say this word now, or underexpressed in a particular cell type. As you can see, the genes that are overexpressed in this cell type and in this other cell type is totally different. So there are some of them that are shared, but most of them are different. So suggesting that we can identify cell type specific responses during regeneration. But the other thing that we can do, apart from comparing gene expression profiles, what we can do is to look at how the cell proportions change during regeneration. As I told you at the beginning, our dropsic is quantitative. Therefore, we can quantify how these cells change. As you remember from before, what I told you is that Planarians, when you cut them, they can regenerate a complete adult animal, but during this process, they don't grow. So what happens is that you get a tiny piece or a head, for instance, the Planaria, and now this head has to become a complete adult animal. And it was not known how this happens. But now we can do this. So what we did was to count the proportion of cells from the different clusters, 2 days after regeneration and 4 days after regeneration, and compare it to what we have before regeneration. So in purple, you have these clusters that are depleted during regeneration, and in green, you have the clusters that are enriched. So as you can see, both plots are very similar, and what we can say is that during regeneration, we increase the amount of stem cells, and some progenitors, especially the neural progenitors, and that makes sense because we cut all the heads of all the Planaria, so we know all the pieces are trying to make a head. But the most surprising part was this. So we identified that parenchymal cells are strongly depleted. As you see, is the most strongly depleted lineage from all the cell type. And these are the cells that were not described previously in any way. And actually, if you see here, both progenitors and differentiated cells become less than half after 4 days of regeneration. And we even validated this using in situ again by counting how many of these parenchymal cells exist in Planarians at day 0 and at day 2. So actually, what we found out is that these cells are probably fueling this regeneration process by providing all the components that the animal needs for regenerating. So finally, I just have told you that we have been able to identify cell type specific responses during regeneration and that we can study how the changes in cell populations happen during regeneration. And this will be the end of my talk. I just would like to say that we made like an interactive app where you can look at these feature plots where you can look at the expression of your favorite gene in any cluster or try to see how it changes during differentiation in any specific lineage or in all the cells at the same time. And we have a GitHub repository that explains how to run all the analysis that we have done here, because this can be applied to any single cell data set that you would have to try to identify how cells differentiate. So together, I hope that I have convinced you that we have built an unbiased and quantitative cell atlas, that we got a consensus lineage tree explaining how the cells from Planaria differentiate, and it's combining both computational and experimental methods, that we have identified gene sets likely explaining the differentiation of stem cells, and that we have been able to study regeneration using single-cell transcriptomics. And to finish, I would just like to thank all the people that were involved in this project, mainly people from the lab where I work. So Nikolaus, our boss, and then we have Jordi, our Planarian expert that now moved to Oxford to start his own lab. Petar, Benedict, and Aris, that is part of the computational analysis. Christine and Sala, that basically were doing all the dropsic runs, and Sala doing most of the Planarian experiments. And to our collaborators in Munich that developed this awesome computational lineage reconstruction method. And to all of you for your attention. Thank you.
Thank you so much. Questions, please.
Hi, thanks for your great talk. I have a question about the new cell types that you have found. So You're saying that it is depleted when planaria regenerate?
Yes.
And is your hypothesis that they supply, like, they use it as energy, as extra food for regeneration? Or?
Yeah, exactly. So during regeneration, the planarias don't grow. They don't have a mouth, so they can eat. So they have to make a whole new animal with whatever they have. So doing GO-term enrichment analysis, what we saw is that these cells are enriched in vacuoles and lipids, so suggesting that maybe they contain lots of, these materials that can actually be burned and produce energy for the planaria. And also, like, their components could be used to make the new cells, like the lipids or the proteins could be reused for new cells. So that's our hypothesis.
Yes, that's very interesting. Do you think there are any analogous cell types in planaria to senescent cells in humans or in like mouse or many other animals? Because they are also kind of involved in regeneration.
I honestly don't know. As far as I know, there are these neoplasts that are stem cells, but they are constantly dividing because they are constantly regenerating all the cell types. So senescent cells, I think that they are more metabolically inactive. So I'm not sure if there are those in planarians.
Yes, that's interesting. Well, thanks.
You're welcome.
It was great. I was wondering if you guys tried to ablate those cells and see if it can maybe inhibit regeneration. So
we didn't do that, but previously there were done experiments that to show that actually these stem cells are pluripotent stem cells, because what you can do is irradiate the planaria, so you produce DNA damage, so the cells die when they try to divide. And what you can do is to transfer a single stem cell, and then this stem cell is going to be able to regenerate the whole planaria. So that's what was done, and we didn't do it here because it was already described how these cells behave.
Yeah, but I was thinking about the cells that supply energy, right?
Oh, no, so in planaria it's very complicated to do knockouts. Well, basically you can't. And we didn't do any knockdown experiments and sequence there. We tried and we failed, so we don't know.
And another quick question I had is that you also do this predictions for different cell lineages, how they produce from stem cells, so you know the genetic programs that activated at different stages. Now, can you? Oh, you probably cannot delete those genes and see if you can ablate the production of those specific cells in planaria.
So that's what we tried to do, but we didn't got enough cells in the end. After doing so, the idea was to do a knockdown of one of these genes that we see that is specifically activated in a lineage, and try to sequence what was afterwards. But we didn't have enough cells in the end to try to assess whether it was working or not.
Thank you.
Oh, it's closer. No, it's no. Equality.
When will be ready analogic work on human body?
I don't know. I think that it's way much more complicated, because you would need to sequence all the different time points during development, right? Because the problem is that if you sequence an adult human, in case that you could, you won't have the progenitors, you won't have enough stem cells. And for all these methods to work, you need to have not all the cells, but a reasonably amount of all the cells in the differentiation stages to be able to reconstruct the process.
But you plan make something else on more complex organisms?
Me personally, no. I'm not trying to do that, but because I think that it's very complicated for the sequencing, and I know that there are already groups trying to sequencing, for instance, mouse development very tightly. Also, there were these zebrafish papers this spring. So I think that people is going towards this way, right? And the question is whether you want a computational method. So the advantage from this is that you can use it for a normal single cell experiment. But I think that also there are many other groups that are trying to develop this CRISPR-based lineage tracing, so which you can reconstruct the the differentiation of individual cells. So I think it's more complex than just sequencing, and I think it's on the way. It will arrive soon. Yeah, as I said, I'm hoping that someone else is doing it before me. They are already working on it.
Just a silly question from me. What do these experiments in Planaria can teach us about regeneration and how it can be applied maybe to human?
Okay.
I know it's the worst question in the world. I'm sorry.
No. So I don't know. So for instance, what we know is that the stem cells in Planaria don't have these classic transcription factors that we have in human, these OC4 and NANOC. So they are not expressed there. So they have a different way of maintaining themselves. Still, there are many other factors that are common to Planarians and other species. For instance, it was known that, for instance, MBNL, that is an RNA binding protein, is involved in the differentiation of stem cells, both in Planarians and in, I think it was mouse. So there are many of the genes that are common and maybe are regulating similar pathways. So I think that we need to start from here and then try to see how much is conserved and how much is not.
Sure, thank you. More questions?
What is the difference between stem cells from Planaria and early embryonic stem cell like blastist went for 30% embryonic cells?
Yeah, so the thing
from
is
human, for example.
The Planarians that we are working on in the lab, they are asexual, so therefore they don't produce sexual cells, so they don't have gametes or reproductive organs. So I think that that's the main difference. But they can produce any other cells. That's why they are not totipotent and just pluripotent, because there's no embryonic development in the Planarians that we have in the lab.
Hi, you are.
Well, it's a short follow-up question on what Alex asked you. I just don't know why is it a problem to generate knockouts of Planaria?
So the technology doesn't exist, but usually when you are doing a knockout, what you're trying to make is a knockout stem cell that you differentiate an animal or a gamete. And there is no embryonic development, so you would have to do knockouts in all the cells, in all the stem cells of a Planaria to be able to do so. And we are not able to culture the cells outside the Planaria. So I think that is just technically not possible now, but maybe in the future it will be possible.
Mm-hmm. Yeah, I see, thanks.
Okay, thank you so much.
Okay.